Chapter 8, Genes to Protein. So by the end of this chapter, you should be able to describe the function of RNA and protein in the cell, describe how the central dogma is used to get from a genotype to a phenotype, and explain the process of gene expression. Okay, so going back a little bit, we talked about 4 macromolecules, right? Proteins was one of them. So proteins are our macromolecules that are made up of repeating amino acid subunits. They have many different functions. For example, you could have muscle contraction. Here's an example of muscle contraction up here, which we don't go too far into just yet. But basically we have actin and myosin acting together to contract muscles. That could also facilitate chemical reactions. It can fight infection. We have different proteins in our body that perform different functions, and some people have different proteins than others. So again, some of the functions of proteins, one of the ones that's discussed in your book, actually I'm not sure if it's discussed in your book anymore, but we're going to focus on it in this lecture, is the antithrombin protein. And basically this protein helps prevent your blood from clotting improperly. We want a little bit of blood clotting, right? We don't want our, if we cut ourselves for our blood, to just keep running and running until we run dry. But at the same time, we don't want our blood clotting so much that it causes something like a stroke. So we need this protein. When it's present in our blood, it helps inactivate enzymes that promote clotting. If you do not have this protein, well, most people have this protein, but it's not functioning properly depending on your genes. So if it's not functioning properly, that's when you get abnormal clotting, and that's when that leads to strokes or heart attacks. So again, our proteins are one of the macromolecules, one of the 4 that we discussed, and they're made up of amino acids. So these amino acids are the building blocks of proteins. There's 20 different amino acids. Remember, some of those are essential. We have to take in through our diet. But basically, they all have the same core structure. So you can see our different amino acids down here. And they have the same core structure right here, but they have different side groups. So the different side groups are what make them different types of amino acids. Again, there are 20 different ones. And basically when we're forming proteins, we make this nice linear strand of amino acids, and then it gets folded up into some 3D sort of protein. And proteins are kind of like a lock and key sometimes. So they each have their own specific linear structure usually, but some of them might have very similar or the same linear structures, but are folded in different ways to make different kinds of proteins. And that can also lead to whether it's functioning normally or not functioning at all. Okay. So these amino acids, what happens is they bond together to form these linear chains. Again, these linear chains fold into this functional 3D shape. That's what determines the shape and function of a protein, is the sequence of amino acids that you get. So again, we might have some similar amino acids that fold into different kinds of proteins, but a different linear amino acid sequence usually comes with a protein with a different 3D shape, and therefore would also have a different function. So you can see the difference between these 2 right here. There's only a few differences, these last 2 right here and this one right here. And because of those differences, it'll fold differently, and it will also have completely different functions in the body. Okay, so where do proteins come from? You guys have heard that you have genes, you have a genetic code, you have a genome, right? And a gene is basically just a sequence of DNA that contains instructions to make or encode at least one protein. So all of the instructions to make proteins are encoded in genes. So your genetic makeup, all of your genes tell your body what the instructions are for which proteins you're going to make and which ones you're not going to make. So a gene is just a section of DNA. Again, it has a nucleotide sequence with instructions to make at least one protein. And then we have something called gene expression. So if that gene is actually expressed, if that gene's turned on, then it means that that encoded protein is produced. So you might have genes to produce certain proteins, but they're not turned on or expressed, which means that your body's not actually going to make them. And this doesn't just apply to humans. I use humans a lot throughout these examples because it's easier. It's easy to understand in human bodies, but this happens in all types of different organisms, all different types of cells. Okay. So we find these genes on chromosomes, right? And each chromosome carries a unique set of genes. Okay. They usually have lots of different genes along their length, and each one contains instructions to make at least one protein. So again, here's a chromosome here. You can see that there's different genes along it. Right here is what we're specifically looking at, the antithrombin gene. And if that gene is expressed, it means we're going to make a nice normal antithrombin protein, which will keep us from clotting abnormally. Okay. So gene expression is, simply put, the synthesis of a protein from a gene. Basically just means that that gene's turned on and it's going to do what it was meant to do in terms of making or following the instructions to make the protein that's encoded in that gene. Okay. So when we start getting into a little bit more with like Punnett squares and genetics here in the next few chapters, you'll see this a lot too. So let's say that a gene we write out as capital A, little a, okay? Each one of these, so this is one gene, but each one of these letters is an allele. An allele is just an alternative version of the same gene. So say this is our antithrombin protein gene, okay? But it's made up of different alleles. Each allele has different nucleotide sequences. And different alleles can influence protein function. So again, a version of a gene with a different nucleotide sequence is an allele. Alternative nucleotide sequences may code for a protein with a different shape. So if we're looking at our antithrombin gene allele 1, okay, so that's our antithrombin gene. This is allele 1. Here's our normal nucleotide sequence. You can compare this with our allele 2, and you can see it's just one nucleotide change. There's only one shift. But when we go to our gene expression, up here on our normal nucleotide sequence, we produce a normal antithrombin protein, which means our blood flow is normal. Down here, where we changed one nucleotide, it's our alternative nucleotide sequence, now we've produced an abnormal antithrombin protein. It has an abnormal shape, and that means that it doesn't perform its normal function. So now we don't have a functioning antithrombin protein, which means that blood flow is restricted, it can clot, it lacks that functional protein to keep the blood from clotting normally. So usually keep in mind, if you have different alleles that distort the shape, or if something else happens to these proteins, if it has a drastically distorted 3D shape, the most likely outcome is that that protein is not going to function properly, if at all. So that's usually what you get. It's not normally that you get something completely different in your body happening. It just means that that protein's not going to function. So these are alleles. Again, just alternative versions of the same gene. So you get, one from mom, one from dad. We'll talk about it more later on, but that's what alleles are. So we all have the same genes, but we have different alleles in all of us, and that's what makes our genetic makeup and our genes and the proteins we express different than each other. Okay. The central dogma of biology. So you'll hear this a lot. Central dogma often refers to DNA, but really what it is how you take a genotype and turn it into a phenotype. So this is how we go from DNA to messenger RNA, or commonly referred to as mRNA, into a protein. And this is what this whole lecture is really going to be about. So a genotype, that little AA that I just wrote, that's a genotype, right? That's a gene that has different alleles, so you're just seeing that it's a genotype. AA, capital A, little a, right? That's our genotype. We want to get all the way to a phenotype. A phenotype would be the antithrombin protein, okay? So this is oftentimes something you can see, oftentimes something you can't see. Let's say that our capital A, lowercase a actually was blonde hair, right? And then maybe capital A is brown hair. So that's the genotype is the AAs that we're seeing. The phenotype is the color of the hair. So again, a lot of times you can see this, a lot of times you can't. Depends on what kind of protein or what gene you're looking at. But in this case, let's say it's antithrombin protein. So no, we don't see it, but yes, we can tell if you have a functioning antithrombin protein by how your blood clots. So we have to get all the way from genotype to phenotype, which means that we have to go through a few different processes that we'll talk about here in just a second. So to get from DNA to mRNA, we have to go through a process called transcription. And then to get from mRNA into a protein, we have to go through translation. So I'll go through another example here in just a second, but basically your DNA, you've got to transcribe it into something, basically into a different language, and then you've got to translate it into the final product. So as an example, let's say that our DNA is the original sketch of a car design that an engineer comes up with, right? That doesn't mean much. If you just hand that sketch of a car design to some guy that you want to make a car, that's not really going to happen, right? You've got to go through a few other steps. So if you have the engineer's original sketch of the car design, the first thing you want to do is make copies of that sketch and turn it into a blueprint, okay? So now we're making it into something that you can actually use, like instructions that you can use to build this car. Then we are going into our next step. So once we have the copies of the sketch that are made into a blueprint, then we go into the next step, which is now we actually have to send that blueprint to the guys that are going to make it, right? So we're sending it out to the factory floor. This is what our messenger RNA or mRNA does, is they're sending this out to the floor so that they can actually start making the stuff that we're talking about. Translation is basically these machines using that blueprint to make the actual car. So now they're translating those instructions into something real, into something that can happen step by step. And then the final product, or the protein, which is the phenotype, again, we may see it, we may not, that's the finished car. So we're going from someone's original idea, right, or DNA, the instructions for the original idea, and turning it all the way into the final product, which, if you're looking at the car example, is the car. If you're looking at the central dogma, we're taking the instructions for what protein we want to make of the DNA, and we're going through a whole process to actually make that protein. Okay, so this whole process here is called gene expression. So again, you might have a gene and it might not be expressed, which means it's not turned on. Sometimes this is a good thing, like say if it were a gene associated with cancer, we don't necessarily want to express that gene, right? We don't want it turned on, we don't want it making those proteins, but there are some, like say antithrombin, we do want that gene expressed because we do want to make those proteins normally so our blood can clot normally. Okay, as a quick refresher, the structure of DNA. So we've got these 4 different nucleotide bases that we're made up of: adenine, thymine, guanine, and cytosine. A and T always go together, G and C always go together. And those are the rules of complementary base pairing. So each strand of these is held together by different hydrogen bonds between these bases. Again, A with T and C and G. Okay. Keep in mind, RNA is going to be a little bit different. But when we're looking at these genes, they have 2 different kinds of sequences. Okay, they're basically organized into 2 different parts. So we have the smaller sequence first, which is known as the regulatory sequence. The regulatory sequence is what determines when and how much protein a gene makes. So it controls the timing, the location, and the amount of gene expression. Okay, so again, it's going to determine when a protein's made. It can turn on or off the gene. It can determine how much of the protein is made, determines in which cells that gene is expressed. This is our regulatory sequence. Okay. The next sequence, which is usually the longer sequence, is the coding sequence. And the coding sequence determines the amino acid sequence of the encoded protein. So this is what determines the actual sequence of amino acids in the protein. So this is what's going to, again, kind of determine what the actual protein is. So you guys have probably heard of genetic engineering, genetically modified crops, like all of that kind of stuff. And genetic engineering, there's positive and negative things about it that you might hear, but overall, we've been doing genetic engineering for a while, and it does, again, have positive and negative side effects, arguably mostly positive. But genetic engineering is basically just assembling new hybrid genes. Okay, so it's novel combinations of regulatory encoding sequences. So basically, you're taking regulatory sequences from one organism and encoding sequences from another to combine them to get what we call a recombinant gene. And recombinant gene is basically just making a hybrid gene that does exactly what we want it to. So if we're wanting to make an antithrombin recombinant gene, that we can ensure that that antithrombin is expressed only in mammary cells, we can use the regulatory sequence of a milk gene, say from a goat. So we're using their beta-casein gene. We're just cutting it, basically, getting those enzymes from the regulatory sequence, putting those over here, and then we're getting the human antithrombin gene and using that coding sequence and adding it to the regulatory sequence of the beta-casein. And now basically what that's doing is it's making sure that antithrombin is expressed only in the mammary cells. So there's lots of different examples of recombinant genes that we can make, and a lot of them have really great medical usages. There's also, food usage for it, lots of different kinds of things, lots of different reasons to genetically engineer these genes. Okay, so let's get more into the detail, right? We went from DNA to mRNA to a protein. So how does that actually happen? So now we've got to go through what is actually happening in the process of gene expression. And again, that's where we're just converting genetic information into a protein. So there's two main steps to this. The first one is transcription. The second one is translation. Okay. So first up, transcription. Very important to remember this. Transcription is happening in the nucleus of eukaryotic cells. But if we're looking at prokaryotic cells, it's happening in the cytoplasm, right? Because our prokaryotic cells do not have a nucleus. So transcription is happening in the nucleus of eukaryotic cells, which is most likely what you'll see for the most part in this class. Is we'll probably be talking about eukaryotic cells for the most part. And it's in the cytoplasm of the prokaryotic cells. Translation, on the other hand, is in the cytoplasm no matter what. Eukaryotic or prokaryotic cell, it's occurring on ribosomes that are in the cytoplasm. Okay, and again, remember when we first talked about ribosomes, they're our spot or our location of protein synthesis. It's all kind of coming together now, right? This is where we're making our proteins and actually synthesizing them and sending them out to do their job in the body. Okay, so transcription. So again, in eukaryotic cells, right now we're talking about being in the nucleus, which is where our DNA is. And the very first thing that has to happen is, remember when we're replicating DNA, this is very, very similar to that, but now we're going to introduce something new called RNA. So in DNA, right, our DNA molecule has to unzip. Our DNA polymerase then goes through, reads, and adds complementary base pairs to it. This is almost the same thing, but it's called transcription, which means that now we're working with RNA. So an RNA polymerase is going to come up here and bind to the regulatory sequence just ahead of the gene's coding region. The DNA strands are going to unwind. It's going to expose the coding sequence of the gene. So this is a little bit different when we're talking about DNA. Usually we have to have the helicase, which is unzipping the DNA, has to unwind first, and then we add DNA polymerase. This is a little bit different. RNA polymerase is going to add first, okay, and then second, it's going to unwind. Okay, so keep that in mind. And then the third thing that's going to happen is now RNA polymerase is actually going to go through, and it's going to add new nucleotides to the RNA. Okay, so it's doing the same thing that it was doing in the DNA, except for now when it's reading the DNA coding sequence and it's synthesizing a complementary RNA strand, we call this RNA strand mRNA or messenger RNA. And it's called messenger RNA because it's going to go from, let's say we're talking about a eukaryotic cell, so it's going to go from in the nucleus outside to the cytoplasm. So this is our messenger RNA. This is the guy that's bringing the blueprints to the floor. Okay. So RNA moves along the DNA strand, and as it moves, it reads the DNA coding sequence. But keep in mind, normally we have A with T in DNA. In RNA now, the only difference is that A goes with U. So in RNA, there is never thymine. It is always U, which is uracil. Okay? So that's the only difference. So if it sees an A, it's going to add a U. If it sees a G, it's going to add a C. If it sees a C, G. U, or sorry, this will actually be a T, because it'll be DNA. It'll add an A. Okay? So you'll still see this in the DNA, but the RNA, you will never see the T's. You'll only see the U's. So it's just going through and adding, just like DNA polymerase did, but now it's doing it with RNA polymerase. And unlike DNA, RNA only has one strand. Okay? So this complementary mRNA strand is going to form from the DNA template according to the rules of base pairing. Again, now we're just pairing adenine with uracil instead of thymine. So as this mRNA strand is formed, it's going to detach from the DNA sequence, and the DNA is just going to reform its double-stranded helix. So it's basically not harmed in any way, right? So the DNA is separating, the RNA polymerase is adding new nucleotides to make its own mRNA strand, and then the DNA is just going to go right back to normal so that it can be used again, okay? So the mRNA is going to go ahead and leave the nucleus. The gene remains part of the chromosome, so all of that DNA stays in the chromosome, stays in the nucleus. The DNA never leaves the nucleus, okay? And we can use it again and again for transcription. So this process can happen over and over and over, which is good, right? If we have antithrombin proteins, we don't just make one and done for our entire lives. We're making them several times. And it depends on what protein and, what's happening in your body for how often you need to make these proteins, but that's all of the instructions that are encoded in your genes. Okay. So again, mRNA is going to go ahead and leave after transcription occurs. It's going to go into the cytoplasm, but our DNA is going to hook back up with each other, form that double helix, and it's going to stay in the nucleus. Okay, so now we've left the nucleus. We have our nice new strand of mRNA, and basically what that's going to do is it's going to have to go through translation. Translation is just using that mRNA sequence to assemble the appropriate amino acid sequence of the protein. Okay, so mRNA is going to leave. It's going to go find a ribosome, our spot of or location of protein synthesis, and it's going to find the ribosome in the cytoplasm, and now we're going to go through translation. So what's happening with translation is that we're basically just going through and reading what our mRNA molecule is. So our newly transcribed mRNA associates with a ribosome. The ribosome moves along the mRNA, reading it. So this is kind of similar to like what we were talking about with our polymerases, right? But what the ribosome does is it tries to make it a little bit more efficient, and it reads it in groups of 3 nucleotides called codons. So you can see here these little groups of 3 like AUG, UCA, UGG. I know it's kind of hard to see me actually doing this, but it's reading it in codons. So codon is just a group of 3 nucleotides, okay? So you can see like AAU, that's a codon. UGC, that's a codon. And it's just doing that because 1, it's more efficient, and 2, each of those codons specifies a particular amino acid, okay? So basically what it's doing is it's taking the very first strip. So we have our mRNA, okay, and let's say it's AUG. And it's reading it in codon. So it's reading all 3 of those at one time, and it's adding an anticodon, okay? Oops. So this is our codon. It's going to add an anticodon, which is just the opposite. And that's what we call our tRNA, or our transfer RNA, okay? So here it would be UAC. Once we have UAC, that's going to find the matching mRNA codon. Okay, so codon here, triplet codon mRNA, anticodon is being added as a triplet code on tRNA that recognizes a specific codon. So we went from AUG in our original UAC, and now we're adding it into our anticodon. So once we've turned the mRNA into tRNA, what happens is the specified amino acid is added to that chain. So the ribosome is just moving along this mRNA here, and as it moves along, it adds a tRNA anticodon, which adds a amino acid, and it just grows this nice linear chain of amino acids. Okay. So it's moving along, we have this cool chain of linear amino acids, and then at the very end, the finished amino acid chain detaches from the ribosome, and then it folds into its nice little 3D shape and becomes the actual protein that we set out to make originally from our DNA. So that's how we go from DNA all the way to a protein. And that, again, is gene expression. So that means our genes turned on and we're actually going to take the instructions and make the protein that we set out to. So remember when we're translating our mRNA into tRNA. These mRNA codons specify particular amino acids according to a universal genetic code. So there are usually, I think in every case, there's usually at least 2 different codes that you can use. Some have as many as 4 codes that you can use for one amino acid. So remember, there's 20 amino acids. You could have any of these 4 codes, and it means that you will always have valine. These 4 codes mean alanine. So it's very predictable. You can always know which sort of amino acids you're taking, and then you can kind of figure out what your protein actually is or what it looks like as well. So again, it's pretty easy to be able to predict the messenger RNA and amino acid sequences from the original DNA sequences. So you can use these rules of base pairing along with the genetic code to predict the amino acid sequence that's going to result from a particular DNA template sequence, which means that you can look at someone's DNA or their genes and kind of figure out what proteins they're likely going to express, which is, how we can start looking for certain genes in people and understanding different diseases, cancer, different genetic defects, or, even really positive things from genetics. Okay, so for your class activity, your Chapter 8 class activity, this is going to be a little bit, well, it's not too bad, but you just have to go through and actually do it. So if you're looking at your class activity right now, you pause me and start looking at it. Looking at the first page, there's a few different steps to this, right? So you're always given the DNA code. Right, so if you're looking at the very first one, you'll see our DNA code, which is GAA. So the first thing we have to do to our DNA, right, is we have to transcribe it. So again, if we're in a eukaryotic cell, we're transcribing it in the nucleus from the original DNA. So we're going to transcribe it, and we're going to make it into mRNA. Sorry, it's really hard to type or draw on this. So we're going to make it into mRNA, so we're going to use CUU. So now we have our mRNA. It's going to leave the nucleus of our eukaryotic cell. Our DNA is going to go back to normal, be ready to use again if it needs to. Our mRNA is going to make its way into the cytoplasm. It's going to find some cool little ribosome. It's going to hook up with the ribosome. The ribosome is going to work its way through and read it as a codon. So this is one codon here, CUU. It's going to turn that into tRNA, which is just the anticodon of this. So C goes with G, U goes with A, U goes with A. Now we have GAA. Once you have the anticodon or your tRNA, then you're going to go ahead and look on this little table here, which is on the second page or the back side of your class activity. And we're going to look for the tRNA code, which is here, 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 or here. Okay. And we're going to look for our amino acid. Okay, so we're looking for GAA right now. GAA is right here. So our amino acid that goes with this anticodon is leucine, or LEU. And the symbol that goes with it, that goes down here, is L. Keep in mind the symbol is just used for this class activity so that you guys can get the ultimate message or phrase that we're decoding. So we have L. And then you can see the example goes on. So it says life, I, and then the rest of it's blank. So you're going to figure out the rest of this. Okay. So let's do the example after I. So the example after I think, is AGC. Okay, so that's our DNA. Now we want to transcribe it into mRNA, so we've got to go, let's see here. Yeah, AGC, so we're going to go A goes with U, G goes with C, C goes with G. Now we're going to take the anticodon, or the tRNA of this. So U goes with A, C goes with G, G goes with C. Okay, so we're at AGC. We're going to find AGC up here, which is right here. AGC is serine, is our amino acid, and our symbol is S. So far we have life is blank. So you're going to go ahead, figure out the rest of that, and then when you turn it over to the next page, or you go on to page 2, then you have 2 more messages that you'll decode. Again, the messages mostly make sense. One of them might be, or both of them might be a little bit strange, but they make sense. They're spelled correctly. So if you get something incorrect, you've done something wrong. So you've got to go back through and make sure that you have each part right. So make sure your transcription is right, then your translation is right, and from your translation, make sure you've got the right amino acid that leads to your symbol. So hopefully it makes sense. Again, a lot of times seeing an animation of transcription to translation can really help you visualize this whole process and understand it. So I definitely recommend different YouTube videos. I know you have some good animations on Launchpad as well, so please go through and understand those, understand what they are, and go through and make sure that you decode each of the 3 messages on your class activity before you turn it in. This might be one that's easier to write out. You can go ahead and write it out with or without printing it, or you can go ahead and type it, but you should have, I need to be able to see each step. So make sure that you write out your mRNA strand that goes along with your DNA code, your tRNA, your amino acid, and then your amino acid symbol. Okay. And when you see the DNA code, it's just going to be a whole bunch of letters, right? So it'll say like A, A, T, G, C, T, A. So I usually go through, especially if you're doing this handwritten, and draw like a big line here to indicate each codon, so you can kind of read it a little bit faster, and you can be like, oh, okay, A. Okay, so you can go through each one a little bit easier. So make sure you break it up into codons. It's easier for our bodies to do it that way. It's also going to be easier for your mind to do it that way. So go through and decipher the messages here. And obviously, if you have any questions, please let me know.